Greetings from Lisie College of Allied Health Sciences.
Good afternoon.
Yeah, today the fourth day of our academic webinar series, Excellentia. So today we have the young and energetic speaker, Dr. Swathy R. She had done PhD in cardiovascular biology, and she is working as SR of Frontier Lifeline Hospital, Dr. Cherian Heart Foundation, Chennai. And she is my beloved and respected friend also. So Dr. Swathy, hearty welcome you.
Thank you very much. Thank you very much. Thank you, Dr. Shabir, for suggesting me to your people so that I got an opportunity to.
Thank you, madam. Before that, then I also welcome my dear colleagues, students, and principal, teachers, and faculties, other professionals from various colleges and industries. On behalf of LC College of Allied Health Sciences, I heartily welcome you all. And now I request Ms. Sharanya, Assistant Professor, LC College of Allied Health Sciences, to introduce Dr. Swathy R. Sharanya, madam, please.
Okay, thank you, sir. Good afternoon, everyone. I am Sharanya VG, Assistant Professor, Lisie College of Allied Health Sciences, Kochi. It is indeed a great pleasure and honor to introduce our chief guest, Dr. Swathy R, to today's webinar series. She will be discussing on the topic regenerative medicine, basics and clinical application. Dr. Swathy R, she is an alumna of Government Victoria College, Palakkad and Calicut University. She had completed her PhD in cardiovascular biology from Pondicherry University. She has participated in various research activities, including decellularization of xenografts from Sri Chitra Tirunal Institute of Medical Science and Technology, animal implantation studies from CMC Vellore, Senior Research Fellow in Biomaterials Development and Stem Cell Isolation from Frontier Lifeline Hospital, and latest being in molecular cloning from AMI Biosciences. Also, she has an excellent experience in teaching profession, and presently she is working as Senior Research Fellow at Frontier Lifeline Hospital and Dr. K.M. Cherian Heart Foundation, Chennai. Without further delay, on behalf of the entire Lisie family, I would like to extend a warm welcome to Dr. Swathy R to this online webinar series. Dr. Swathy R, Senior Research Fellow, Frontier Lifeline Hospital and Dr. K.M. Cherian Hospital Foundation, Chennai. Welcome you, ma'am, and proceed with your topic.
Thank you, Sharanya, ma'am. It was indeed nice hearing you talk like that. And I think, yeah. So hope all of you can hear me. Okay. Yeah. Good afternoon, all. I think it's very nice, and I take it as a privilege that I get an opportunity to talk to the stars of Lisie Hospital, of course. And like within all the disappointments and the hardships of this COVID-19 pandemic, I think because of that only I got an opportunity so soon to meet you all and share something what I know, which may help you in future definitely. And I'm so excited, also equally wondering how you would take the regenerative medicine topic because it's totally out of your syllabus. Being a zoology background person, I had no idea that I would come up to this cardiovascular biology or do something like this. It's not my subject knowledge, but the passion took me here. So I think you people would be more into medical field. I had zero idea about what's happening in the medical side, and I think if this comes to you, you can analyze it in a medical side, and you can suggest better procedures or protocols to improve this. At least 3 or 4 of you would get inspired, I think, as I was. When I joined for my PhD, I wanted to do something on markers or something. But my mentor, Dr. K.M. Cherian, he had great influence on me. I had wondered what the people over there were doing in this tissue engineering. And I could develop a passion. I read something. I think I had been doing something good. But let's see. Hope this will help you also. This will take you to think out of the box, out of your textbooks. And maybe some of the Nobel Prizes are going to come from Lisie Hospital this batch. And I would be taking it very great pleasure to start this topic. I hope you all will get some information from this, and I would share whatever I know.
Madam, please, sorry for interruption. ഒന്ന് കുറച്ചും കൂടി വോളിയത്തിൽ സംസാരിക്കാവോ? നിങ്ങൾ ഫേസ്ബുക്ക് ലൈവിൽ അത് കിട്ടുന്നില്ല എന്ന് പറയുന്നത്. If you don't mind.
Okay. 1 minute.
ഓഡിയൻസിന് കേൾക്കാൻ പറ്റുന്നുണ്ട്, പക്ഷെ ഫേസ്ബുക്ക് ലൈവിൽ പോകുമ്പോൾ ചെറിയൊരു ഇഷ്യൂ, അതുകൊണ്ടാണ്.
Hope this will work. Ekamo, can you hear?
ഇവിടെ കേൾക്കാൻ പറ്റുന്നുണ്ടായിരുന്നു. FBയിൽ പ്രശ്നം. Okay, you carry on. Okay, sorry.
Okay, no problem. So regenerative medicine, personalized medicine, recently we are hearing too much about all these, especially when something like a pandemic come up. So regenerative medicine is a translational research branch of tissue engineering and molecular biology. Translation. So DNA to RNA is transcription, RNA to protein is translation. Anything is expressive when it's translated. So that's why we are calling regenerative medicine a translational research from bench to bedside. So regenerative medicine is nothing to be learned or practiced, but it should come up as a treatment for everyone, those who are in need. This is a translation research that's combining tissue engineering and also molecular biology. This is used to replace, engineer, or regenerate the cells. When something happened to an organ or a tissue, we must replace it or we must repair it and also help it regenerate to form a better new tissue. So adana regenerative medicine kondu uddeshikkunnathu. This is a very promising field because it not only repair a damaged tissue but also help it to function within the host body. നമ്മൾ ഒരു ടിഷ്യൂ ഒരു ഹോസ്റ്റിലേക്ക് ഇംപ്ലാന്റ് ചെയ്യുമ്പോൾ വാട്ട് ഇറ്റ് ഹെൽപ്സ് ഇസ് അത് തനിയെ ഫങ്ഷൻ, ഇറ്റ് മസ്റ്റ് കോൾ അപ്പോൺ അതേഴ്സ്, അതിന്റെ നൈബറിംഗ് ടിഷ്യൂസിനെയും സെൽസിനെയും ഒക്കെ വെച്ചിട്ട് ഇറ്റ് ഷുഡ് ഫങ്ഷണലി ഹീൽ ഇറ്റ് സെൽഫ്. ആ ഹോസ്റ്റ് ബോഡിയിൽ അത് ഹീലായി ആ ഹോസ്റ്റ് ബോഡിയോട് ചേർന്ന് അതൊരു ഫോറിൻ മാറ്റർ അല്ലാതെ ആവണം. ദെൻ ഓൺലി ദർ ഇസ് യൂസ് ഇൻ റീപ്ലേസിംഗ് or transplanting anything to a host. Regenerative medicine is a combination of 3 things: biomaterials, tissue engineering, and stem cells. Rationale for development. So anything doesn't get invented or discovered on its own. There must be a reason. Rationale means there must be a reason so that people wanted something to come up. ഇതുണ്ടായാലേ ഇത് സോൾവ് ചെയ്യാൻ പറ്റുള്ളൂ എന്നൊരു ആവശ്യം വന്നു. That's a rationale for anything. So natural thing is aging. All of us get aged. What happened in aging? Telomeres get shortened, cell death സംഭവിക്കുന്നു, oxidation, protein misfolding, DNA damage, all genetic things are varying. Naturally that will affect the organs. Aging ആവുമ്പോൾ organs get damaged. What we do when any organ gets damaged, we go for a transplant or replacing. It can be aging, it can be any particular disease, it can be infection, it can be trauma and injury like accidents or anything. We lose a particular part of an organ. So what we need to transplant it. അങ്ങനെ ഒരു നീഡ് വന്നപ്പോഴാണ് we went for the development of regenerative medicine. I think there is a lot of voice disturbance. So transplantation, Dr. Shabir?
No, you can carry on.
Yeah. So with the problems of transplantation of organ and tissues are not in the order in the PowerPoint. The main thing is lack of donor organs. We can see the transplantation list in the websites. you can see there are a lot more people waiting and dying due to lack of any donor organs. Maybe it's a problem of genetic matching or donor availability, the cost factors, the waiting time, all of these. Suppose if at all you get a donor organ, what happens? We are given immunosuppression drugs so that the foreign organ is, the organ received is not considered as a foreign organ by our body, and it gets adjusted to our own body so that there is no rejection. Adananu we take immunosuppression drugs. Then there must be genetic matching, പിന്നെ organ preservation techniques ശരിയായിരിക്കണം. So all these bring us to a clutch that transplantation is not really very easy, not at all easy. You can see the graph. Leave the other countries. You can see India. This is the organ donation rate here. So much people are only getting organs. If it's organ donation, definitely it is the organ receiving. So only so much people are receiving organs. So there is a very essential, there is a need of a very essential alternative to this organ receiving or organ donor, donating issues. So I would not say this has come to us, but this is coming in the near future. Tissue engineering will have a lot to do with this to solve these issues. So hope you understood what was the rationale. So transplant or replacing, it may not be transplanting the whole organ, but any part. Take it a heart. You know, after a heart attack, what happens? There is oxygen deficit to a particular part of the heart. So that part becomes inactive because of oxygen deficit. It becomes inactive. It has a color change. It turns black, or there's a scar. So though we clear the clots, clear the blood vessels, the affected part remains the same. So the idea behind tissue engineering approach is, we are making a tissue to replace that damaged part of the heart so that the tissue becomes the heart's own part. If we can't keep anything and everything there because it will get rejected, it will get calcified. So this is the history. So long back, started something like tissue engineering without knowing what it was really. The term was coined in 1987. Then a lot of works had been going on. This is Robert Langer. He is called the Edison of medicine because he had brought about so many milestone experiments, so many really valuable research that could come up to clinical applications. So he is believed as the father of tissue engineering. What is a tissue? All of you will be knowing. This is just a slide, an introductory slide. Microenvironment plays a vital role in maintaining differentiated function and gene expression of cells. So tissue is not just a group of cells, it's a group of cells. Then the cells secrete many things, which becomes the extracellular matters, growth factors, hormones, all together form a functional tissue. Yes, the last one. These tissues better mimic the in vivo physiological state as compared to monolayer cultures. Monolayer cultures, നമ്മൾ cells ഒരു layer ആയിട്ട് grow ചെയ്യുന്നു. അപ്പോൾ അതൊരു group of cells ആണ്. But that cannot be called a tissue because a tissue must be functional. Cells ഉണ്ടാവണം, അത് cells ഉണ്ടാവണം, അതിന്റെ secretions ഉണ്ടാവണം, ഗ്രൂപ്പ് ആയിട്ട് ആ എക്സ്ട്രാ സെല്ലുലർ മാട്രിക്സിൽ അത് ഒതുങ്ങി നിൽക്കണം, അത് ഗ്രോത്ത് ഫാക്ടേഴ്സ് പ്രൊഡ്യൂസ് ചെയ്യണം, അതിന്റെ ആക്ഷൻ വേണ്ടി ഹോർമോൺസ് ഉണ്ടാവണം. Then only it's becoming a functional tissue. Now tissue engineering. You can see in the picture a biodegradable polymer scaffold in vitro tissue culture the cells. Yes, you can see the methodology. Then I'll go back to that slide. So tissue engineering la ee factors ഒക്കെയാണ് വേണ്ടത്. ഒരു biomaterial scaffold, then cells, growth factors, or a bioreactor. So what is a biomaterial scaffold? Scaffold means something that can hold something. അപ്പോൾ ഇവിടെ ടിഷ്യൂ എഞ്ചിനീയറിംഗിൽ നമ്മളൊരു സ്കാഫോൾഡ് വേണം, അതിൽ വളരാൻ സെൽസ് വേണം, ഗ്രോത്ത് ഫാക്ടേഴ്സ് അല്ലെങ്കിൽ ബയോറിയാക്ടേഴ്സ് വേണം. അപ്പോൾ ഒരു ബയോമെറ്റീരിയൽ സ്കാഫോൾഡ് എന്ന് വെച്ചാൽ എനിത്തിങ്,
Excuse me,
it cannot
madam.
be anything.
Excuse me, madam. Excuse me,
Yes.
madam.
Yeah.
You can use English. Some participants are from the outside state also.
Definitely.
Better to use English.
Okay. So tissue engineering is involving biomaterial scaffold, cells, growth factors, or a bioreactor. So what is a biomaterial scaffold? A scaffold is something that can hold something else. So a biomaterial scaffold in tissue engineering means something, a membrane that can hold cells onto it. The membrane is not same for anything. It differs according to the requirement. For replacing a hard tissue, biomaterial would be something else. For bone, it's different. For cartilage, it's different. Then for anything, depending on the tissue to which we are into or on which we are trying to do a replacement or a repair, the biomaterial ways. Then the cells. Cells naturally, what we are using now is stem cells. And easily isolation can be done in the mesenchymal stem cells. It can be taken from any adult organs or muscles or adipose tissue, fat and all. Then growth factors or bioreactors. If we can give growth factors into this, it will be helping, it will enhance the cell attachment, cell growth onto the scaffold very nicely. For that, we are giving growth factors. Bioreactor is something which give an environment. So we have a scaffold, we have cells, we have growth factors. But we need every organ develop in a different environment. So we need to create an environment, an artificial environment, so that these cells can easily attach to that scaffold. These growth factors can induce its action on these cells and the scaffold so that it develop into a functional tissue. For that, we use a bioreactors. Now the parameters in tissue engineering. The cell source, we can't take any cells to do the tissue engineering. It must not cause any rejection. It should be grown in culture. It must be active and it must be controllable. Anything beyond control is a tumor. So it must be controllable. We must know the environment where it can start the division, how we can stop the division. So there must be a control. Scaffold. Look at that, the structure. The structure of the scaffold must change according to the requirement. For which organ we are trying to replace or fix the new biomaterial, according to that, we should develop scaffold. Say for bone. For bone, it must be very hard, isn't it? Because it must be strong. The same cannot be with the skin or with the heart or with any other tissues. So for skin, it must be very elastic, Then for heart muscles, it must be strong. It must have so much strength. It must take in so much pressure. So the structure of the scaffold selected must be different, will be different. Then the logistic template, it must be biodegradable. When we implant a biomaterial, we don't expect to be in that organ forever. What we expect is the biomaterial should go into that organ. It must be there. It will have cells. Then it will initiate angiogenesis. That is, the cells will be there. It will say heart. Now, I will always, what I know a bit is about the heart. I would talk about that. So imagine we are making a patch after a heart attack or say. Then what we do? We keep the patch there, where it is seeded with cells or not. So what we expect is this patch with the cells must interact with the environment. It will face the fluids of our body. then we expect the growth factors to move into it. These cells must attract other cells to come to the scaffold. We expect those cells also to attach to the scaffold. The scaffold get the blood vessels, it gets vascularized. And gradually what happens, the scaffold goes down, it gets eliminated, but that area is recellularized or repaired with very new hard tissue, our own tissue. So that's why it must be biodegradable. In the other case, if it's not biodegradable, after some time what happens? It stays there, it gets calcified, it causes infections, or it releases a lot of carbon dioxide, causing necrosis. Then there must be a custom design. Yeah, depending on the going to implant it, there must be a custom design. Sometimes it must be more porous. Sometimes it must not be that porous. It must be a bit opaque. Then bioreactor, as I told earlier. Cell seeding. Cell seeding is the technique by which we take cells from the particular source, immunocompatible sources, and we put them onto the membrane, and we are letting them grow. So seeding. Seeding means giving cells to these membranes so that it forms a layer over the membrane and get attached to it and start differentiating. Then environment. So cells grow in a particular temperature, carbon dioxide limits, so that should be maintained. Regulatory signals, and if that needs this bioreactor, sometimes they give small vibration so that it will keep the tissue under tension, the scaffold under tension, so it takes up more and more cells. So that's the duty of bioreactors. So the cells must be immunocompatible, they must be growing in a culture, they must be active, and also we must be able to control them. The scaffold, it must be the structure related to the required organ. It must be biodegradable, and also the design must vary. The porosity or tensile strength, pressure, standing powers, it must vary. And bioreactor, of course, it helps seeding cells onto this, whatever scaffold we have created. So that's what the biodegradable polymer scaffold. It is taken in a petri dish, or say, cell plates. Then we are seeding cells onto it, whatever we want. So you can see bone, cartilage, liver, intestine, ureters. So whatever kind of cells we want, we can seed the tissue with those cells, the scaffold with those cells, making it the scaffold and the cells make it a tissue. Those tissue we can go for in vivo implantation. So these are the important things we need for tissue engineering. The novel chemistries, the new growth factors. Every day there are more and more findings. The biomechanics, because not just cells and the tissue, the scaffold would be a tissue, but we will be giving some external pressures or some mechanical forces. Then the decellularized organs, 3D printing. It's a very interesting field. 3D printing, we can print the organs in the desired size to make testing or to create new organs. Also, cells. The scaffolds currently used. There are a lot of synthetic scaffolds and also biological scaffolds. I've given some synthetic scaffolds here: PLLA, PGA, PLGA. The effects, I would brief. It will reduce the bioactivity, degradation rates. So they may not be biodegradable always. So that will cause the problems, what I was telling earlier. They'll try to degrade themselves. They'll release a lot of toxic chemicals, and this will cause necrosis. If they are degrading, it will damage all the surrounding tissues. Then we'll have to replace it. After operation, we'll have to do a reoperation and explant the graft and replace it with a new one. Then in cardiology, there'll be a structural deterioration. The implanted things will get calcified, the leaflet will tear, then adverse immune responses will be there. Not only the transplanted tissue gets damaged, but also the neighboring tissues would be getting affected. And of course, they are exorbitantly high cost. I will not say all of them. For bone replacement and all, they use hydroxyapatite and tricalcium phosphate. They are synthetic, but for bones, they are very good so that they can hold, they have the strength, the porosity, and hold things in position. I'm talking about the soft tissues. So these are the heart valve failures. Calcification, wear and tear, thrombus. Now, why we are suggesting biomaterials when it comes to transplantation? The biomaterials, as I said, is biological scaffolds. They are taken from living organisms. Earlier, when it comes to transplantation, we were using homografts. That's from cadavers. We used to take the tissues immediately. We used to store them. But that's also not reliable because we may not be able to take them easily, or the numbers would be very less. So for biomaterials, we are using animal tissues. In our case, we were using bovine and porcine, buffalo, and pigs, and even fishes were used as biomaterials so that they'll have structural and functional similarity to the native tissues. They'll have the structure. Say it heart. In Frontier Lifeline, what we are using is heart tissues from bovine. So there are patented techniques. It's an approved procedure. And we are now harvesting all these from certified abattoirs. So we take heart pericardium and process them. So when it comes as a heart patch, when we are trying to replace the damaged tissue with this patch, it has totally the same type of structure to what our heart has, and also functional similarities. Then the extracellular matrix. In a synthetic scaffold, it'll be just a porous membrane. Then we are giving cells, we are giving growth factors. But in a biomaterial, if we are taking from a buffalo or pig, or say humans, there are autographs that we are taking from our own body. In that condition, what is it? That biomaterial itself is an extracellular matrix. We need not give anything extra. So what happens when this biomaterial is processed? It'll induce cell division on its own. Then chemotaxis, that is, it'll enhance the neighboring cells to come to it. Then there is interaction, the contact between these cells and the tissues. From that, what happens? It differentiates into that particular organ. Then there is constructive tissue remodeling. So when a biomaterial is implanted, naturally what happens? Our body calls for the immune response. Our body is under pressure. It wants to know if this is a good material or if this is not matching our body profiles. So what happened? There is a remodeling. So there are two types of remodeling. It can be constructive or it can be destructive. In destructive tissue remodeling, what happens? The immune system identify the new material as foreign and harmful, and it will send a lot of macrophages. And in both conditions, the first reaction would be the same. All macrophages, giant cells, all these immune agents would come. Then gradually, they'll stay with the tissues, and they'll understand it. They'll see what is released from these tissues, what is the action. Then if the tissue is good, what happens? It doesn't affect it. It turns it into a constructive tissue remodeling. These macrophages, with their own help, they convert the tissue implanted into a constructive tissue. They help the degradation of this implanted tissue and make it the host on tissue. Then it stimulates in vivo migration proliferation of cells. It will help the cells to come and migrate onto this tissue. Then formation of a biointegrated, a whole tissue. Then it's hemocompatible, it's durable. It's a ideal homograph alternative. It is just like a human tissue when it has the ECM, when it has cells onto it. So what in the latest, now what they do is they take from our studies, we take cells from that particular person, We grow them in the labs, then we collect those cells, seed onto the scaffold, and then keep it back to the patients. So what happens? The patient is receiving nothing new, a scaffold that is totally processed, and the cells are from the patient's own, that is autologous. So it becomes more or less the same as the person's own tissue. Then, yeah, of course, it mimics the physiology and mechanics of the native tissue. After my PhD work, this was the process biomaterial for implantation. We had made out of bovine pericardium. Uh, yeah. So anything, say we are taking a pericardium, we can't use it like that because it will have its own buffalo cells. It will have other antigens that can elicit immune response in the host body. It will have all other extra things. So what we have to do is we must send this biomaterial through a process, through a protocol where all the cells on this biomaterial, all the antigens, everything is removed. And this biomaterial is left with only the collagen bundle, extracellular matrix, and some elastins. So, a tissue is made of collagen. So, we are removing all the cells and other, and there are a lot of techniques, patented protocols for this. So, we must make sure that before any implantation, the tissue we have taken is free of the cells. We are taking a tissue, we are sending it through certain procedures, certain chemicals, then we are making sure all the cells in that tissue is removed, and the tissue is just a collagen matrix and the ECM, the extracellular matrix, so that it is a fresh scaffold like anything, like any synthetic scaffold or porous matrix. It is just like a piece of paper, we can say. Then what we do? We try to create some gaps within the collagen bundle so that there is space for cell adhesion. Then we seed them with cells. We keep them in the incubator or bioreactors. We enhance the environment so that the cells get attached to the tissue. And then only we can implant it into the patients or use it for the studies, anything. So any tissue you are taking, we must make sure it is totally decellularized. The process is called decellularization because all the cells are removed from that particular tissue. Yeah, you can see this. This is just demonstration pictures. Say this is a decellularized scaffold. This is a medium. So we have cells in the culture flask. From the culture flask, we are, trypsinating and taking, we are counting cells, then taking so much of cells in very concentrated suspension. We may not take this much and all. So very concentrated suspension of cells we are taking. We are loading it onto the tissue so that we may not take in so big plates because the plate must be filled with the tissue so that the cells would have no other place to attach, or the plate can be non-adhesion plates so that the cells don't attach to the plates. So cells naturally migrate, move, and attach to the scaffold we have given. So we are keeping it, we are changing the medium in adequate intervals. We are keeping them in incubators or bioreactors. And this is a very advanced picture of a lab-grown organ, kidney. These are all under research. It's not come to a real translational stage. This is for your better understanding. I've taken this picture. So by giving stem cells, and all the cells develop into a particular organ in different environments. So if you are giving environments and factors which help it turn into cardiomyocytes, the cells on the tissue will develop into cardiomyocytes. And if you are very lucky enough, they start beating, and it will be very interesting to do all those. If you want a kidney, you want nephrocytes, you can give the stem cells, you can give the particular environment and growth factors which will help these cells to differentiate into kidney cells. So that will be developing into a part of kidney. And this one we can take, we can use 3D printing to create a kidney structure. We can keep them in the medium. We can send stem cells through that, and we expect that tissue, kidney shaped structure to develop into a vascularized tissue. So what are the clinical applications of tissue engineering? We have already come across, I think, reconstruction of tissues, bone regeneration, cartilage replacement, nerve graft, skin replacement, cardiac patch replacement. There are many. I'm just mentioning few. This video was very interesting. You can see the image there. One of an army nurse and some, I think in US, she lost her ear, her left ear, in some accident, and she wanted to, but she wanted ear there. So this doctor, what he did was he made a cartilage to the form of to the shape of an ear. Then he implanted it along with the growth factors into the forearm of that nurse, that person who wanted the ear, and he let it grow for 8 months, and he harvested it. The skin lost from the forearm was replaced from the thigh skins. Then he took this and he has surgically fixed that ear onto that lady. So this is the recent thing I've seen, but they're waiting for the results. So they are expecting results. They have done the surgery, few months back. So they are still expecting results to see what happens after a particular course of time to this explanted graft after surgically fixing onto the ears. It's not that easy. Doctors will know the procedures, that there must be vascularization, blood vessels, something around. But I have idea only about the tissue engineering and making of this ear. Yeah. Burn injuries. This tissue engineering can help a lot in this burn injuries. You can see there is numerical details about how many people get burn injured in India, how many deaths, how many accidents. So in this case, tissue engineering will definitely help. In Brazil, what people do, in the burn wounds, they keep tilapia skin. They don't process it, they just wash it, they laminate it, and they just, if hand is burned, they just keep that tilapia skin over the hand, and they do some dressing, and it is seen that the patient's tissue grow. There will be a difference, and the due course of time, this tilapia skin get peeled off, but the recovery is very soon, and the function is restored very soon. So in tissue engineering, recently what we were doing in Frontier Lifeline was we were using fish skin, that marine fish skin. We have processed it. We have made into a particular form, and with the help of with the advice from CLRA, the Leather Research Institute, we could develop it into a better a membrane or a sheet, and we expect it would create breakthrough effects in this burn injury. Already there are people in goat skin or buffalo skin. They're taking it, they're decellularizing it, they are treating it properly and keeping over the burn wounds of people, so that the skin, which the processed biomaterial what we are keeping on the skin, it's expected to degrade on its own, but by the time it degrades, our skin will develop a fresh new place in that burn wound. It is really of great importance. I strongly believe in future this must be a very breakthrough research, and for burn injuries is the best thing, because you need not go surgically. This is external application, so it can restore the function, initiate or accelerate wound healing. It will reduce pain because it's not, the wound is no more exposed, and it's achieving the desired cure, and also skin is regenerating within. So this must be very helpful in burn injuries, I believe. So the frontier medieval tissue engineering, what I was talking about all the time. So these were what we were using: buffalo, pigs, pericardium, fish skin. You can see This is the native cells in the tissue. We are decellularizing it. Then there are no more native cells. Those red rounds are the cells. There are no more native cells, but only the collagen and fibronectin. The collagen and all these things are necessary for keeping the ECM or the membrane in an intact manner. So those will be retained without any damage. Then we are using it in clinical application like valve replacements or whatever. So decellularization. Decellularization, we have a lot of techniques like detergents. Then some people do it mechanically. But thing we have to keep in mind is after decellularization, only the cells and antigens must be removed. There must not be any harm occurring to the native architecture of the tissue, of the scaffold. Then only we can use it in the correct manner. We can handle it properly. If the collagen fibers are broken, if the collagen bundles are so much separated, it will not have so much, it will affect its elasticity, its physical strength, all those. Now, stem cells. This is the history. In 1960, hematopoietic stem cells were discovered. Then embryonic stem cells from mouse blastocyst. This embryonic stem cells from mouse, it was of great help because in this blastocyst, when these ES were isolated, it opened to a huge area of research because we can use this murine models, blastocyst models. They could modify the genome of rats. I mean, they could modify, take some cells, modify cells, then inject them back into the blastocyst so that the adult mouse will be developing with a required or desired feature which we were looking for. So embryonic stem cell of mouse blastocyst was really a breakthrough. Then the other one was Yamanaka's. He got a Nobel Prize in 2012. What he did was he took skin cells called fibroblasts and converted them into pluripotent stem cell by altering the expression of certain genes. Now, what are those cells? I'm sure all of you must be knowing the stem cell types. Totipotent cells, the embryonic cells. It can develop into anything. Pluripotent cells, then that's again cells of the blastocyst. It can also form any cell type. Multipotent cells, it can form tissue cells. So these are the sources. Now there are embryonic stem cells and adult stem cells. So adult stem cells word is kind of misleading. It's not that only adults have it. The kids after embryo, it becomes a baby. Since then, it has adult stem cells on its body. In our blood, say the heart, bone marrow, adipose tissue, everywhere we have stem cells. But thing is, the numbers are very low. In bone marrow, we have stem cells which are producing the blood cells. In adipose tissue, we can harvest stem cells. Even in the heart, there are, cardiac stem cells. First, we were thinking the heart is a completely differentiated organ. But later, But later it was found out that even the heart has stem cells, cardiac stem cells, adult heart. But the numbers are very less. So what we do when there is a problem, when there is an end-stage heart failure, we inject stem cells taken from that patient into the heart walls. So the expected result is these stem cells are injected again. So they now start multiplying. They help or they, inspire the already resident cardiac stem cells in the heart to grow and develop and make the damaged part functional. So that's what's expected. But there are a lot of glitches in the stem cell therapies. Still, only we can verbally tell it, but it will take. There are a lot of challenges in that. So the thing is, all these organs are having adult stem cells. From them we can isolate and harvest and isolate stem cells. So this is from my PhD work. That's a white vista rat. We are taking adipose tissue from that using forceps after anesthetizing, and the tissue was processed. It was chopped. It was treated with collagenase. It was broken down, then centrifuged, and we get cells like this. These are the shape. This is the shape of mesenchymal stem cells. We can characterize them with fluorescent this FACS technique with certain markers. So day 2 it looks like this. Day 5 it looks like that. Day 7 and day 8 confluency set in. Confluency in the sense that the flask in which we were growing the cells has been totally covered with these stem cells. So that is just an example, a demonstration, so that you might feel at least a bit interested that something is really there. So we have taken this adipose tissue, we have made into harvested and made them into the stem cells. So these are the potential uses of stem cells. All of you must be knowing there is wound healing. These are all in the future. I may not say this is in the near, this is happening or stem cells can cure so and so, but very few. The problem is there are a lot of challenges in this field. There must be proper regulations. We must use it properly. There must be limitation in the numbers. There must be limitation in the way you are, and there must be guidelines in the way you are injecting these stem cells onto it. And if the numbers are high, we know what happens. There will be tumor, there will be uncontrolled growth, unnecessary growth, factors will be released. So stem cell therapy is still, it's yet to come. It's not always success. It has a lot of issues. So these are the challenges. So embryonic stem cells are isolated. We are growing in vitro, that's in the laboratory. We are growing a zygote. From that, we are taking embryonic stem cells. But a zygote developing in vivo and in vitro is different. So naturally, anything developing in vitro will not have all the features, all the advantages which is developing within the body. That's the difference between God's creation and man's creation. So God's creation is mighty. It has everything in it, but not like ours, because we have limitations. We are yet trying to do something, but we are not established or accomplished the full result. So embryonic stem cell in vitro, we are trying to develop in the laboratory on Petri plates. So it will be a zygote, but unless it has the environment within the human body, or it has the factors moving in and out, it will never be the same as an embryo. So there is high likelihood of it being rejected by the body. So it cannot be always accepted by the body. So that is a major drawback. Then what happened? Some people find it morely troubling to use stem cells derived from embryos. So some people believe ethically, they believe it's like killing the embryos. So killing an embryo is like killing a life. So ethically, there are a lot of issues in bringing about in vitro embryonic stem cells. Then pluripotent stem cells. What are pluripotent stem cells? That Yamanaka got a Nobel Prize in 2012. So he was trying to find out something else. So it's a serendipity. So pluripotent stem cells are anything which can grow into a particular line of cells or a tissue. So what happened was he took adult stem cells, skin cells, and he tried giving different factors. What happened was when the 4 particular factors were altered, the stem cells reverted back. From embryonic, it's coming to adult stem cells. These adult stem cells were again taken back to their embryonic stage. That is called pluripotent stem cells. But it's not practical to grow it easily in a laboratory. So researchers are investigating ways to improve the process. If it's possible, we can develop many organs, tissues in the laboratory. But the protocol is very, delicate. The factors, everything is very difficult to achieve in a laboratory. That's where I thought you are all medical students, medical people, so you would know better about how to treat something, what can be done, what is to be altered. If you go into biotechnology or molecular biology, I think it will be an asset for the growing medical field. I was a zoology person. I'm a zoology person. So I had my own limitations to understand what it is. Only by going with them, standing near them, listening to what they say, attending their talks, I got some idea about what's happening. But I think it will be much easier for you guys, so that some biochemistry about this, the medical side. So I expect some of you would come up, some of you would bring a breakthrough in this pluripotent stem cell research, and I'd be very happy that at least give a 1% of your thoughts or your ideas to it, or at least you will plan that, okay, we must do something on this. Then adult stem cells, as I said, there are stem cells in all adult organs, but they are found in relatively very low quantities in the body. There is only, we say, a niche, a small group of adult stem cells, whether it's in the bone or muscle or heart tissue. It's in very, very less quantity. So there is a greater chance that they could contain DNA abnormality. So we don't know what is the feature of that cells. So when they emerged, it was a particular feature. Maybe they are inside for a long time. Maybe their genetic constitution must have changed. The DNA structures must have changed. So there is a need for detailed studies to infer how embryonic stem cells develop, so that they can understand how to control the type of cells created from them. So embryonic stem cells we can develop, but we must know, once taken out, how we can control the growth, how we can help it to differentiate in the desired way as we require, then how it must work, when and where it must be stopped, so that it doesn't develop with an adverse effect. So these are all the challenges. So stem cell field is vast, is very promising, but the thing is, we must face all these challenges to come up with a special therapy. And there are a lot of regulation. We have to register with different bodies. They'll give us guidelines. Not anyone and everyone can do this. We see a lot of, I would say, fake advertisements of hospitals. We are doing stem cell therapy on this, that. We must always look for the license. We must try to understand if it's true. We must not just blindly believe them. So, yeah. So these are the references. If anyone of you want to know in detail, because we have time constraints, and I'm not an expert, I'm also a beginner. So I am also learning. I've shared whatever I'm sure about and whatever I know with you. And if you want to know more details, and if you're interested, you can take out all these, these are the important things. I know Stephen Badilak is the main person who started off with decellularization. He's the person who compared the techniques. So it will be helpful if you can find his article and read. So I'm thanking my mentor, Dr. K.M. Cherian here. Thank you.
Thank you. Thank you, madam.
Yeah, it's okay.
So it's a time for question and answer session.
Definitely.
If anybody have any doubts, you can unmute yourself or raise your hand by using the option which is given in Zoom app. and ask the question. Otherwise, you can put your doubts in a chat box also. I think one doubt is there from Rishi. Swati madam, can you hear me?
Yes, yes, I can hear.
One doubt is from Rishi. Can you explain the relation between stem cell cultivation and non-human cell cultivation?
Stem
As
cell
you said,
cultivation?
yeah, as you said for biomedical implantation.
Yeah.
That is a question.
I didn't understand. Stem cell?
Can you explain the relation between
Isolation?
stem cell cultivation and non-human cell cultivation? That is what he mentioned. Rishi, you can
Okay.
unmute and you can ask the question. You can explain.
Yes, yes.
Rishi.
Ma'am,
Yes.
good afternoon, ma'am.
Yes.
Yeah, I just want to know the difference between stem cell cultivation and non-human cell cultivation, which means the cells cultivated from other than humans like animals. How do they do the process in separate us
No,
for stem cell
earlier,
cultivation?
yeah, earlier the stem cells were cultivated from animal proteins. From animal
Yeah,
proteins, then growing them, giving all the factors and all. But that is not was not immunocompromising. Yeah. So that was like we are giving the foreign, there was this rejection because it was not suitable for human use.
okay, ma'am.
So yeah, there are a lot of studies mentioning this cultivation of stem cells, but better than taking it from the animal proteins and cultivating as human cells, we can. That's why we have come up to this adult stem cells, because easily now we are taking from adipose tissue. Adipose tissue is not a useful tissue for us. So as an alternative for those stem cell cultivation techniques, we have come up to adipose tissue harvest and taking mesenchymal stem cells from them. So I would say it was not much of practical relevance. It had a lot of issues with that.
Okay, ma'am. Thank you, ma'am.
Yeah.
I have a doubt. I have a question. Sir, Shabir sir.
Yeah, madam,
Yeah,
you
yeah.
can. It's a Dr. Deepa from Lisie College of Allied Health Sciences.
Hello.
Hello. Actually, I have two questions. The first one is regarding the slide you showed where you have that heart culture, cells you are isolating that from heart and how it is grown in the lab. So my question is it really viable? Because for skin organs like skin, which are mainly structural, of course, function is there, but structural organs like skin and all, we can, I feel it is convincing. But this heart, which or as a matter, kidney or any such organ, is it really viable to grow such a complex organ in the laboratory?
It is viable, ma'am.
Commercially, is it any commercially any commercial thing is there? I mean, is it any product is available?
No. That's what I said. This is just a demonstration picture. So we have patches. There are a lot of commercially available patches for heart developed from different porcine and bovine pericardiums. We are attaching, we are growing stem cells, we are seeding them with stem cells and attaching, we are suturing into the heart muscle, the affected part. So we expect it gets the chemotaxis and it becomes myocytes. And also we have grown, in our lab, we have grown cells, we have plated them, then we have given them microenvironment of a cardiac cells, and my seniors, not myself, and they have seen it beating within the plate. So that's happened. There are membranes, but this is just demonstration. Nobody has developed a heart or nobody has transplanted, to my knowledge.
Yeah.
But it will come up in future, definitely.
Long way, but
Because,
heart
yeah,
and
long way, definitely.
It's a complex organ. It's not just structural. It's a functional organ. Even kidney is a functional organ. So that this is a long way. So the next question is
that's what
3D
printing. 3D printing, we can make a structure. We can with a desired material, we can make a hard structure. Yeah. Then we are passing cells through it, where fluids rich with fill with cells. We are exposing them to a microenvironment of developing cardiomyocytes, and we expect finally it will beat once. But there are a lot of lab-grown heart and kidneys and all functional, but it's never tried on humans. Definitely not.
And next question is, you said decellularized scaffold from animals, no, porcine, bovine. What we will do for no zoonotic diseases? They will be harboring virus, no. Some disease will be there for animals. So how we can
First, as soon as we harvest itself, we test the samples. We have kits. We test all the samples for any zoonotic, diseases. This virus kits are there. So we take not only the pericardium, we take blood samples from them. Only cardiac surgeons go to harvest these. So they take blood samples with them for each animal and each pericardium. They test for any zoonotic disease. If anything is found, we'll just discard the pericardium. If we bring, say, 120, only 20 we'll be using.
So if
but
we
from
want
20 pericardium, we'll get really lot of patches.
So if we have a product, like from this scaffold from animals, and if we have a product, do we need some any other regulation? No, wouldn't it be
Definitely. Yes. Now in Frontier Lifeline, they are almost 11 years. I just joined in between. 11 years till now. When we finished the last trial, we went to Delhi for approval for the multicentric trial to the CSIR. So it's 11 years, I must say, that they can't sell it as a product. But already 900 plus patients in our own hospital with better results. We have submitted all the results to the DCGI, ICMR, and everybody. And they asked to do the multicentric trial. It's to AIMS, PGI Chandigarh. To these hospitals, we have to supply our tissue. Their surgeons must use this. They must give a feedback. And after so much feedback, only we can bring it as a product. It's a long way. Now, but there are less restrictions. There are a lot of easy regulations to go. But long back we started, so we are following the same guidelines from them.
Thank you, ma'am. Thank you.
It's okay. It's nice.
Thank you. Any more questions or feedback or suggestions? Anybody? Okay, I feel there is no more doubts or questions from anyone. Hence, we are moving to the end of the session. I invite Miss Sajida. She's a BCVT first year student, bachelor degree in cardiovascular technology. I invite her for delivering vote of thanks officially.
Hello everyone, I am Sajida Kia, first year BCVT student of Lisie College of Allied Health Sciences. As we are approaching to the end of this session, I feel highly honored to propose the vote of thanks on behalf of Lisie College of Allied Health Sciences. Firstly, I would like to start by thanking Reverend Father Cherian, Assistant Director, Lisie Medical and Education Institution, and the Lisie management for providing all the support for conducting the webinar. Next, I would like to thank Dr. Swathy, PhD in Cardiovascular Biology, Senior Research Fellow, Frontier Lifeline Hospital, and our distinguished speaker of the day for her very much informative session on regenerative medicines, its basics, and clinical applications. Thank you, ma'am, for your valuable information.
You're welcome.
Next, I thank our dearest principal, Dr. Shapira Sikbal, the mastermind behind the session for managing and conducting a program like this through a digital platform and making it a huge success. Thank you, sir. I also like to thank all our beloved teachers, non-teaching staffs, and students of Lisie Medical and Education Institutions and all other audience for their active participation throughout this meeting, making this a huge success. Last but not least, I would like to thank the IT department of Lisie who made this meeting possible. Once again, on behalf of Lisie College of Allied Health Sciences, I thank each and everyone who participated in this session, providing that even we are far apart from each other, but our minds are closer, and we shall overcome the deadly pandemic. So thank you everyone. Have a great day.
Thank you, Sachita.
Thank you all
So,
for listening to me. Hope everybody will think about this, and all those challenges will be ruled out by you guys. Wish you all a great success. All of you stay safe. Thank you. Thank you,
thank you.
Dr. Shabir, for referring me. Thanks a lot.
Thank you. So today's session is ended. Thank you all the dignitaries and participants, especially today's speaker, Dr. Swathy. So hope we all can meet the next session. Thank you. Thank you all.